Good day. In today's lecture, we will be discussing about biological membranes. Now, this is the materials will be coming from Fundamentals of Biochemistry, 5th edition, by Donald Vowet, Judith Vowet, and Charlotte Pratt. However, the chapter itself, we won't be discussing the whole chapter. We will only be focusing on the biological membranes. So, key concepts for this lecture is certain amphiphilic molecules that form the bilayer, and the bilayer as a fluid structure in which lipids can rapidly diffuse laterally. So, basically, we have here a structure of a micelle. So, lipids can come into many different forms. So, you have here, especially phospholipids, as you all know from your um, biochemistry subjects, you have your polar heads and hydrophobic or nonpolar tails. Now, we have lipids that can that has one tail or two tails. So, one type of lipid is the one having one-tailed lipid. So if you look at the structure of a single-tailed lipid, you will see that this is the hydrophobic head, which is relatively larger. This is the polar head. And then this is the hydrophobic tail, which is the a single hydrophobic tail. And if you look at their shape, it is a conical. It produces a triangular, or basically in a three-dimensional view, this is a conical shape. And this conical shape is actually much preferred when they form structures such as this, which is a micelle. So a single layer of lipids, whereas all of the tails are concentrated inside and the heads are outside. The reason why your lipids form this is because of the hydrophobic effect. The hydrophobic effect is the tendency of nonpolar substances to expel water or to aggregate together against the water. So basically, in an aqueous environment, such as the inside of our cells, inside of our body, the polar heads of the phospholipids are the one in contact with the outside. Sorry, are the one in contact with the outside, whereas the hydrophilic tails are the one inside. So this is slightly larger um, phospholipid. So you have here, this is another one. So sometimes the micelle, you have a water trapped inside a micelle. So this is another configuration. Again, this is a single layer because the conical shape, for the conical shape, it's much easier to form a single layer micelle. However, since this is a polar, water is polar and it is interacting directly with your hydrophobic tails, there is a certain amount of uh, high energy or rather instability in this region because again water doesn't want to have anything to do with the hydrophobic tails and the hydrophobic tails doesn't want to have anything to do with the water, so they tend to form a slightly oblong shape um, version. As you can see here, the single-tailed lipids really have an effect because you have this sort of niche. It can't really form bilayer. And that is why for membranes, stable membranes, the optimum version or the optimum form is actually having two tails. So the two tails or the having two tails for a lipids prevents the shape from being conical, but rather it becomes more cylindrical. And having a cylindrical tail, it becomes easier to form bilayers. So here is an example of a liposome. This is a vesicle inside of your cell. And stretching that image, this is your membrane. This is the phospholipid bilayer. As you can see, two-tailed lipids formed end-to-end in a bilayer configuration. So let me show you a bit about an animation of how your bilayers form. So as you can see here, an animation whereas you have your bilayer. So I will play this without the sound. The full video is available in your Blackboard. So anyway, here is your bilayer. So again, they are not covalently linked with one another. And you have your moving, they are actually very fluid, they are moving. And the one of the things about bilayer is that because of the hydrophobicity of this inside areas, hydrophobic molecules can also intersperse within that. Although, for example, here, cholesterol is a molecule, a hydrophobic molecule with a hydrophilic side, which is the OH group. Now, the cholesterol is important in bilayers because it promotes its stability. So it prevents too much hardening of the bilayer at lower temperatures, and it also increases, or rather it increases the fluidity of the membrane at low temperatures, so it prevents the bilayer from freezing easily. And it also at very high temperatures, it also stabilizes the membrane, so it allows them to stick more closely together and not disperse at very high temperature. So cholesterol here can intersperse in your lipid, so they can insert themselves inside. So the OH group, the hydroxyl group here is interacting with the polar heads. Now here is your phospholipid bilayer simulations in water. So the gray areas here is the water. You have the orange one, the hydrophobic tails, and then the colorful hydrophilic heads. As you can see, your membrane lipids spontaneously form bilayers in an aqueous solution. So the gray balls here are your water. So this one here is important because for membranes, it allows a self-sealing capability to the membrane. So if it becomes punctured, your membrane can seal itself, just spontaneously seal itself by just or rather through hydrophobic interactions. So as you can see now in this structure, so from the video animation or simulation video showed a while ago. So you have here this structure of your um, bilayer. So as you can see, they are freely moving. So we have two types of movement in your phospholipid bilayer. So one is the transverse diffusion, which is what we call flip-flopping. So that means that one phospholipid flip-flops or moves to the other side of the bilayer. So for example, from the outside to the inside or inside to the outside. So this occurs spontaneously, or rather slowly, through a facilitated transfer, or it's facilitated by an enzyme called the flipases. But this is a spontaneous, or rather a non-active type of transport. So you don't really have to spend ATP for this type of movement, and that is why it's also very slow. Because again, the movement is generally energetically unfavorable. So you have a slow, although they occur. What drives their occurrence is basically the concentration gradient. So it depending on because we have different types of phospholipids in the bilayer, so the concentration gradient can drive the flip-flopping, but they are very slow because again, the polar heads must transverse across the non-polar interior of the bilayer to get to the other side, and this is again facilitated by a protein called the flipases. Now the other one, which is a rapid movement, which is the lateral diffusion, just basically your lipids, phospholipids, moving through or along the same side of the membrane. So they just move along. And that is also why they're very rapid. They occur because each individual phospholipid, they are not really covalently bonded with one another. They're just interacting through their hydrophobic or van der Waals interaction. So this is again a very slow movement. And the other is a very high. Okay. So another thing about the lipid bilayer is its phase transition. When we say phase transition, basically the lipid bilayer transitioning from a very fluid to a rather rigid phase. So basically this is mostly due to the temperature. But several other factors can contribute to the transition temperature. So the transition temperature of the lipid, which is the temperature at which your membrane from being fluid becomes a rigid membrane, basically it freezes, is due to several factors such as the degree of polarity of your tails, or rather the degree of unsaturation of the tails. So when we say unsaturation of the tails, the kinks on the tails of your lipids, because of the double Remember, this is a fatty acid molecule. So you have long chains of hydrocarbons. Now, when you have kinks or the double cis double bonds in your hydrocarbon or the fatty acid chain, so those are the kinks. So your lipids have these hooks or kinks that actually makes them more dispersive, so it increases the fluidity. So the more kinks in your membrane, the more the higher, or rather, it requires a much lower transition temperature for it to freeze. So it doesn't freeze easily. So it becomes more fluid. So also cholesterol interspersed in this membrane. It prevents these molecules to easily solidify. So those are also some of the factors. Even the length of the fatty acid chains contribute to the membrane fluidity. So those are the factors affecting the membrane fluidity, and in connection to that, the transition temperature. Because the more fluid the membrane, the lower the temperature need to be for the membrane to transition from a fluid state to a more rigid state. So that's it for the membrane, the characteristics of the membrane. So in the next part, we will discuss about what is interspersed with our membranes, specifically the membrane proteins that are in conjunction or in coordination with the membranes.